All right, good morning, everybody. My name is Steve Minnie, and I'm the VP of Business Development in the Materials Group of Bruker. And I'd like to welcome you to this installment of the Bruker FM webinar series. This presentation is titled Multidimensional Imaging with the Opterra Multipoint Scanning Confocal, and is presented by Jeff Stuckey. Before I introduce Jeff, I'd like to make a few quick logistical comments. First, we encourage your participation in the webinar. And if you have any questions during the webinar, please type it into the questions panel. We'll bring all the questions together and group them and then answer them all at once at the end. Often we have more questions than we have time for, and if that's the case and we don't get to your question, we'll be sure to follow up and answer it by email. Also, if you'd like to review the presentation or pass it on to a colleague, the webinar will be posted online, usually within a day or two. That's at the webinar section of the webpage. And also as a follow-up to this webinar, you'll receive an email with that link. Finally, when you exit the webinar, you'll be asked to take a brief survey. And I would very much appreciate it if you take the time to complete that survey, as it really helps us pick the topics that are most important to you and generally make the webinar series better. So let's get started, and let me introduce our presenter, Dr. Jeff Stuckey. Jeff got his PhD in neurobiology from Cornell Medical College and has spent the past 24 years in the bioimaging industry, including 15 years at Universal Imaging and Molecular Devices before Jeff joined Bruker. He's currently the marketing manager in our Fluorescence Microscopy business unit, which was formerly Prairie Technologies. And he's been doing that for the past 2 years. And so let me turn it over now to Jeff for his talk titled Multidimensional Imaging with the Opterra Multipoint Scanning Confocal.
Yeah, thanks, Steve, and welcome to all the attendees. The topic today, as Steve mentioned, is multidimensional imaging with the Opterra Multipoint Scanning Confocal System. The agenda for the outline for the presentation, we're going to go over some Opterra design overview slides, basically covering light path and scan modes, and some information about the performance of the system, and then we'll get into some application data specifically related to multidimensional imaging. The Opterra really from a design standpoint, was designed specifically for live cell applications, so people typically work with the system with cell cultures or small organisms like C. elegans or Zebrafish. You see a picture of the system here. Complete systems comprised of a microscope, typically an inverted microscope. The blue device here is the imaging scanner. Laser launch, we use a visible laser launch, up to 5 lasers. Standard camera, which you can see here is an Evolve Delta EMCCD camera, and then the instrument rack with the computer and monitor. One thing I wanted to, this is a slide that I got from Paul Maddox this summer. He said it was the concept was really developed, as far as he knew, by Jonas Dorn and Gaudenz Danhauser, the iron triangle of imaging. I think anybody that's been doing light microscopy for any length of time understands this implicitly, which is that there's always trade-offs in microscopy between speed, intensity, and spatial resolution. And one of the advantages of the Opterra, as you'll see as we go through the presentation, it really allows you, it has the flexibility to really allow you to pick where you are in that triangle depending upon the application requirements that you're dealing with. So a quick design overview of the Opterra. There's 2 modes of operation. We can either operate in a variable pinhole linear array field scanning mode, gives better XY resolution, or a super video rate high-speed slit scanner, so that for people that have really high-speed applications, they can sacrifice spatial resolution in one axis, but get acquisition rates up to 1,000 frames per second. We're typically using an EMCCD camera as the detector. One of the big advantages of this system is the high-frequency scanning, and the overall optical design helps minimize phototoxicity and photobleaching, which is something that anybody that's done live cell work knows is always a constant struggle in terms of being able to minimize those 2 factors and get the duration of an experiment that fits in with the application requirements. And it's especially challenging with multidimensional experiments where people are acquiring, typically acquiring Z-series over time. And I think a lot of you know that even though you may, with a confocal system, you're focused on a particular focal plane, there's still constantly illumination above and to some degree above and below that focal plane, which just contributes to the photobleaching of the overall sample. As I mentioned before, the multiple scan modes allow for optimization of speed, resolution, speed, and light delivery. And big benefit of the optical design, which I'll show you some diagrams in a minute, is that the linear pinholes, when in pinhole mode, and the separate light paths for emission and excitation minimize emission crosstalk and reflection of the system. This is just an overhead view of the Opterra imaging scanner showing the light path. This is a very compact unit. In this diagram, the blue line traces the excitation path through the scanner and out to the microscope, and then the green line traces the emission path returned from the microscope out to the detector. The light enters the system through an fiber optic coming from the laser launch, and there's some beam shaping optics in this portion of the scanner that shapes the beam, round output coming out of the fiber into a rectangular beam shape, then passes through, when in pinhole mode, passes through an array of micro lenses. When in slit mode, this array is popped out of the light path. This square or rectangular object here is the aperture plate. On the blue line, you can see passing through the lower part of it, green line passing through the upper part of the aperture plate that's arrayed in this photograph. So there's both excitation and emission apertures on the system, so it is descanned. And you can see, because they're both on this plate, that the alignment between the two is maintained when the aperture plate is shifted from one array to another. Light then reflects off of some optics, passes out to a galvanometer. It's got a reflective surface on both sides of it. So on the excitation path, the light reflects off the galvanometer, and then off of a piezo, mirror on a piezo, which then reflects out to the microscope. On the return path, the light passes again, bounces off a couple of reflectors back through the emission pinholes, and then is reflected off of the backside of the galvanometer and off of another piezo that reflects the return emission to the camera and paints the image on the camera chip. Just a close-up, this shows the area, pretty packed area here where there's a galvanometer and the scope side and camera side piezo. This is just a picture of the aperture plate, and you can see on the right side are the excitation apertures. On the left side is the emission apertures. You'll notice they're on different levels here. Talk about that more in a minute, but we do separate the two paths, the excitation and the emission path, in terms of vertical orientation. They are separated when they come back into the scanner and pass through the dichroic. Different aperture positions that are available on the system. There's a 60 micron pinhole, 45 micron pinhole, and 30 micron pinhole, and then 70, 50, and 35 and 22 micron slits. And this is one of the advantages of the system, is that you can pick the appropriate aperture for the application requirements. So if the application requires better spatial resolution and the user wants to use a pinhole to get better both XY and improvement in both X and Y resolution, they can pick a pinhole that matches the numerical aperture and magnification of the objective that they're using. This is all just done electronically by just making a selection in the software. The aperture plate itself is controlled by a servo motor inside the scanner, so switching from one pinhole to another does not require any kind of physical change of anything in the scanner. Same thing's true of the different slit sizes. Most people typically use like a 22 or a 35 micron slit for higher speed applications. We occasionally have people that are using a 50 or a 70 micron slit. if they're working with an extremely high-speed application. We do have some customers that are working with specialized detectors and running at 1,000 frames per second. They're typically using some sort of a slit, maybe a 50 or 35 micron slit for those kind of applications. This just shows a more of a schematic of the light path. As in the other diagram, the blue line shows the excitation path. You can see it passing through the aperture, excitation aperture being reflected off the galvanometer here, which is scanning back and forth, reflected off of a polarizing beam combiner. We won't be talking about that today, but that's actually plays a role in the photoactivation component of the system. We'll have a presentation on that in a couple of months. Reflects off of a dichroic that is typically notched to provide reflection for the laser lines that are in the system. The blue, like the excitation light, then continues out to the microscope. The return comes back, passes through the dichroic, reflected off the galvanometer, up a couple of mirrors back through the system, off the galvanometer on the back side, and then passes through an emission filter out to the camera. And just to give an idea of how the galvanometer and the piezos work together, basically the galvanometer acts in a similar fashion to an X galvanometer and a point scanning system. So it's sweeping the 32 beams of light, when in pinhole mode, the 32 beams of light that are formed by going through the linear aperture array. Those sweeps those beams of light in a X direction across the field of view on the system. And then the piezo is analogous to the Y galvanometer in a point scanning system. So it sweeps one time per frame, translating the beams in the Y direction. And because there's 32 pinholes, the Y translation only has to cover 1/32 of the field of view, which is one of the reasons that the system's so much faster than a point scanning system. And just to sort of animate this and show you what this looks like, so the piezo moves the beam in the Y direction, one sweep per frame, while the galvanometer moves the beams back and forth across the field in the X dimension several times during a exposure time or a frame. And putting these together, you see what the scan pattern looks like. And this is bidirectional, so the field scanning is in both the galvo sweeps and the piezo are bidirectional to make the scanning as efficient as possible. For people that have an interest in timing patterns, this just shows a schematic, electronic diagram of the waveforms that are being generated. You can see that the piezo sweeps one time per frame, while the galvanometer is sweeping several times per frame. You'll notice that we're in the standard scan mode. We're using a scan pattern for the galvanometer that's linear across the field of view during exposure. We also are blanking the laser during the time that the galvanometers are turning around to sweep back across the other direction. Another important aspect of the system, because we have a linear array of pinholes, 32 pinholes arranged in a 1D array, that the crosstalk in this system is improved quite dramatically compared to a multi-point or a two-dimensional array, something like the spinning disk system or some other types of multi-point scanning systems. The reason for this, and it's actually at least one-half of the crosstalk of a 2D array or less, and you can see schematically here the reason for that, where one pinhole can receive, in the linear array, one pinhole can receive crosstalk from neighbors only in one axis, whereas in a 2D array, any given pinhole can receive crosstalk from multiple pinholes in various directions in the 2D array. And the benefit of this is that a 1D array provides improved axial resolution, improved sectioning, as well as providing better penetration into thicker specimens. Just a quick word about general performance of the system. Using the standard camera that we use with this system, which is the Evolve Delta made by Photometrics, that has a 512 by 512 full frame acquisition, 512 by 512 pixels, and we can generate up to 40 full frames per second when we're in pinhole mode, and we can get faster acquisitions from a smaller region of interest, for instance up to about 500 frames per second with a rectangular ROI. In slit scanning mode, we can go up to 66 full frames per second at 512 by 512, and with a specialized detector such as a Evolve 128 camera, we can achieve speeds of up to 1,000 frames per second in slit scanning mode. So just some general benefits for multidimensional imaging, based upon some of the performance benefits that I previously mentioned here. As I said, Crosstalk provides improved axial resolution and better imaging depth. The high acquisition speeds provide excellent temporal resolution. You'll see some examples of that in a minute when I show you some application data. And that's true of both just basic time-lapse experiments where people are running at a single focal plane at a maximum rate of speed, but as you'll see that in combination with the piezo focusing device that we use, we can achieve high acquisition speeds in four-dimensional data acquisition paradigms also. And the shorter exposure times results in lower phototoxicity and photobleaching. Just a couple of examples based on experiences we've had with customers over the last 8 or 9 months when we've been out doing demos and workshops. This is a great example. Investigator at Marine Biology Labs this summer was working with sea urchin embryos, and their basic paradigm was to do a Z-series every 10 minutes or every 2 minutes, and the Z-series was typically 50 to 60 planes, and they were running their experiments for over 2 hours compared to about 1 hour they were able to achieve in their previous experiments on spinning disc systems. They've been using a spinning disc system for a number of years at the institute that they're at, and they were quite pleased that they were able to run longer-term experiments this summer. We've also found just at across the board demonstrations we've been doing this year that people comment that have had experience with spinning discs, that they're generally seeing exposure times on our system that are 5 to 10 times shorter than they're seeing on spinning discs. So the shorter exposure time, obviously translates into lower phototoxicity and photobleaching, which is always an advantage when you're working with live cell applications. This is just an example, a couple of examples here of imaging range on the system. First one, sorry about the jump there. This first one is a zebrafish that was labeled, the neurons were labeled GFP, and this was 100 micron Z range in this experiment. And this is just showing a three-dimensional reconstruction of the acquired Z series. The next example is a little bit more challenging specimen. This is a tumor slice that was labeled with CFP and GFP for another kind of application. And I was able to get some slices from the investigator that was doing these experiments. They were actually doing the experiments in a multi-photon system, which typically offers much greater imaging depth than a confocal system, but we were able to image this specimen, and again we had 100 micron Z range, and we'll do the 3D reconstruction here so you can see that we're able to image through a relatively dense kind of tissue preparation with success down to 100 microns with this system. Some more physiologically oriented kind of application examples here. This is a time Z series collected using zebrafish embryo as a specimen. So it's, what we're seeing here is a spinal cord in a zebrafish embryo, and this was labeled with Rab5 GFP vesicles. And the data collection here, we were doing 12 sections for the Z series. The Z series were collected in less than 1 second, and then a Z series was collected every 5 seconds. And play the movie here for you. You can see the vesicles moving through the spinal cord here. This is a particularly, this is data collected almost 2 years ago. In fact, it was one of the first experiments I saw run on the Upterra a couple of years ago at Loci at the University of Wisconsin. And this was a particularly interesting experiment because the investigators that came by had been trying to do this on a standard point scanning system, and the time required to collect one Z-series was just too long. On a point scanning system, it was taking 15 or 20 seconds to collect a single Z-series, and that was just too slow for them to be able to visualize what they wanted to be able to see in the experiment. And they were able to visualize these vesicles moving in 3 dimensions with the data they collected on the Upterra that day. And they were quite excited about it. So it's one of those kind of things when you have a chance to show somebody a system running and they're able to visualize something they hadn't seen before, it's always pretty exciting for everybody involved. Another example to show here, the previous example, we were looking at a small organism and at a specimen in which the specimen, the portion of the specimen they wanted to look at obviously existed in more than a single focal plane, just due to the anatomy of the object they were looking at. Even in cell monolayers, which is a very common live cell application, obviously, people are increasingly interested in being able to collect volumetric data rather than a single focal plane. And this example of some data collected this summer or this spring at the Marine Biology Lab AQLM course. These were Hela cells in which they're EFG labeled EB3 end-binding protein, and then H2BM cherry label in the nucleus. And this was done collecting 21 plane stacks with a half-micron spacing. Took roughly a little over 1 second to collect each Z-series, and then doing 5-second intervals on the Z-series. And you can see here the movie. This is the maximum projection of the time points, seeing these comets moving around from the end-binding protein labeling. And this was kind of interesting because with a 3D, I think it's a good example of with a 3D representation of the cell, you have a much better visualization of the end-binding protein label moving through the cell than you would in a single focal plane. And with the speed at which we're acquiring the data, we're able to get a time point every 5 seconds here. Certainly, fast enough to be able to capture the dynamic events that were being visualized in this particular experiment. Another example of a time-based series using cell cultures. This was using Hela cells. This is going to show max projections of Z-series with there was labeling of mitochondria with YFP and labeling of peroxisomes with cherry. In this particular case, there were 11 planes spaced 0.3 microns apart. Again, this is in 2 colors, so there was 1.2 seconds to acquire each Z-stack, and then a Z-stack was being acquired every 2 seconds. And again, you can see with being able to capture the entire volume of the cell, it was quite easy to be able to capture the movement of the peroxisomes and the mitochondria in the cell. And again, this was something where the person doing this experiment had been trying to do this on a point scanning system, and they just weren't able to capture the data fast enough to be able to do the visualizations they wanted to, and they quite easily captured it with the Upterra system. One of the other things that people do that can kind of fall into the rubric of multidimensional experiments is being able to do stage montages, both in just X, Y, and also in X, Y, and Z. And besides the speed with which the Upterra can acquire data, one of the things we've done is developed a special application module for being able to collect these kind of data sets called Atlas Imaging. And I think anybody that's tried to do one of these kind of applications can attest to that one of the challenges is being able to easily set up the data acquisition so that you acquire what the portion of the object that you want to be able to acquire easily. And also to be able to, something we've been able to do with the application I'll show in a minute is to be able to allow people to only acquire data from the area where there's actually specimen available, or specimen apparent. This just shows what the basic interface looks like for Atlas imaging. On the left, you see a small image window, which is actually a live image window, which we'll see a couple of movies in a minute, shows how this works. And on the right side is this workspace that you see, which in this case shows a preview that's been done of a zebrafish, and then showing the grid, the basic grid that would be laid out by having picked a couple of tiles around the zebrafish and asking it to draw a complete grid that would capture the entire area required to visualize the entire structure. This little movie here is going to show how you can navigate using the Atlas window. So what you're going to see is a live image here, but then you'll see also in this thumbnail, you'll also see the same live image and see how as the navigation occurs moving in the X or Y dimension, that this navigation window actually shows you in the workspace area, the tile actually shows you where you've moved with relation to the specimen. And when you find a place that's of interest, you can actually just click on the add or update location, and it will freeze a thumbnail in that particular location. And then as you continue to move around, navigate around to find other locations, that tile will remain in location so that you can see the relationship between the where the current live image is and other places where you've already added or stored as a location. Changing focus has also been simplified here, so that there's on the live image window you can see there's these small thumbnails that allow you to help visualize where you are in Z with respect to other locations in Z that you may have already set, top and bottom of the focal plane. And the control of the Z can be done either through moving, using the mouse to grab on the thumbnail and move it up and down, using a mouse scroll wheel, using the existing controls we had in the software previously, these sort of standard up and down arrows, and also just the wheel on the manual controller on the focusing device. Okay, this is another, I mentioned before, one of the things that we've added to the software is to be able to economize on data acquisition. So you'll notice in this particular example, we were looking at a zebrafish, and here's the zebrafish, and there's a lot of blank area that would be defined by just letting a system define a two-dimensional array of tile locations to be acquired. In this case, we can actually turn off tiles that we don't want to acquire, and this just shows us the user going through and picking the remove tiles feature and go along and click and remove the tiles they don't want to acquire, so that when the system acquires data, it's only going to be acquiring data in the area where there's actually sample. And I think this is another area where people that have done this kind of work in the past can attest to the fact that there's often a lot of time taken up by acquiring what's essentially blank space. So by adding this kind of functionality to the software, we've helped people economize the workflow of the data acquisition of these kind of experiments. Also might mention that we do the same thing in Z, where you can actually define, instead of having a single Z range for the entire specimen, you can actually define 2 or more Z ranges over the specimen area, and then the system will interpolate between those Z ranges you've defined to allow a more efficient acquisition of the Z dimension also. Just a couple of examples of some data acquired using the Atlas imaging functionality. This is the Arabidopsis plant. This is actually a stem. And this is a 2x4 montage using a 60x objective. So not a particularly large one as montages go, but the people that were looking at this specimen were actually quite happy with the rate, the speed of acquisition, because again, this was something he had been trying to do on a confocal, a point scanning confocal, and it would actually take, I think they told me, it would take up to 30 seconds to acquire something like this, whereas they were able to zip through this acquisition in a matter of just a few seconds with the Upterra system. It also benefited from the fact that this area that you see here as background, we were able to turn off acquisition using the pruning function that I showed in the previous slide. Another example, this is from a tissue section. This is a large acquisition. This area shown in the image is actually 2.4 millimeters by 1.2 millimeters, acquired at 60x. So this was, in terms of image frames, this was 18 by 9 images. So it's close to well over 100 images, close to 180 images. This was definitely something that people have been trying to do this on a point scanning system. It was taking a really long time to acquire these kind of data sets, and in this particular case, we were able to run through this acquisition with the Upterra system, including being able to leave out this area that's in the black background area here. We managed to run through this, I think, in a matter of like 20 or 25 seconds to acquire this data set. So they were quite pleased with the improved workflow and throughput, being able to use this kind of functionality. So in summary, with respect to the Opterra multi-point scanning confocal, we provide the flexibility and aperture selection to meet the experimental needs of a user to be able to balance speed, resolution, and sensitivity, so the user can determine where they want to be in the iron triangle of imaging. Pinholes in a 1D array minimizes crosstalk and improves axial resolution compared to 2D arrays like spinning disk. The scanning pattern with pinholes minimizes phototoxicity, which allows people to conduct longer-term experiments or experiments where they're imaging at a higher frequency than they might previously have been able to do. The speed provides high temporal resolution in general, but especially with respect to 4D imaging. You can acquire four-dimensional data sets at temporal resolution with each individual Z-series that is many times faster than people have been able to achieve with point scanning systems previously, and certainly is on par with what people often collect in terms of just a 2D image across time. And then the higher throughput on the system in grid collection applications provides a lot of benefit for those kind of applications in terms of speed and overall work throughput. So that's our presentation for the day, and at this point we'd be happy to entertain any kind of questions anybody might have.
All right, great. Thank you very much, Jeff. We have a number of questions here, and I think we'll be able to get through all of them. If you have a question, pop out the questions box in the control panel and type it in, and we will be able to answer it here or get back to you by email. So let's see. We have a number of questions, two main categories, really sort of on live cell application components and then microscope components. So I guess we'll start with the live cell. But there's maybe just to start it off, there's a general question that the videos through the net were very jumpy. Is there a place that you can see them online or otherwise, or how can people get access to clear video sequences?
Well, they'll be able to watch the webinar again, but we'll also upload the presentation to the website.
Okay.
So they should be able to download the presentation and watch it.
Okay, that sounds good. Okay, so in the live cell imaging, what's the longest that you've ever imaged a live cell?
The longest we've ever imaged a live cell, we've actually done, I don't know about, in terms of experiments with cell cultures, probably the longest one I've seen run was on the order of an hour, and that was just the experimental protocol the person wanted to work with. We've actually done overnight imaging with small organisms. For instance, we did a recent demo at a recent workshop at Harvard, and one of the guys there had a couple times came in and did zebrafish imaging overnight, where he was come in at the end of the afternoon and with the zebrafish, and he was collecting Z-series, about 60 or 70 focal planes every 5 to 10 minutes, and he ran it overnight. Did that a couple times. I've also done experiments with people. We did, zebrafish experiments for a couple of hours. The people at Harvard or NBL this summer were running these sea urchin experiments for about 2 hours every day. So, sort of a general overview.
So sort of along the same lines, how do you maintain cell viability? And I guess that's a question about the hardware setup to do that.
Okay. What we're using on the demo system is a stage-top incubator made by Ocolab. We had some European customers had some experience with it, they liked it, so we've got those on the demo systems. They seem to work quite nicely. I mean, certainly that's something if somebody has their favorite kind of stage-top incubator, it will probably fit in the stage that we typically use with the system, which is an ASI stage. ASI has a number of different stage inserts, and the stage-top incubators made by any company are typically fit into a sort of standard size insert. We've had a couple of people that have also done full microscope enclosures. In those cases, they've either done it themselves, or we've done a couple of systems where we've worked with Beat Luden in Switzerland, who does custom microscope enclosures.
Okay. So this is a little tangential, but how does the system compare to light sheet microscopies for long-term live organism studies?
Well, they're really different kinds of technology. I'm not sure we can compare the type of images that you would get from the two systems are probably. I've never actually, I've never seen data compared side by side of what we collected on Upterra to a light sheet. I think they'd be relatively comparable. And the kind of organisms that people have, that typically work with for light sheet, they're doing long-term experiments. Like I said, we've done things overnight with Upterra successfully. I know some people with light sheet sometimes run things for up to 2 days. I guess it's best with the level of specificity of the question. That's about what I can say about it.
Okay. I think you sort of answered this, or at least alluded to it, but is the system compatible or with an inverted microscope configuration?
Yeah, that's the typical configuration. That's what most cell biologists use. We've also done integrations on upright microscopes, which tend to be people that are more neuroscience oriented, looking at things like brain slices.
Okay. And then sort of also now moving maybe a bit into the configuration, for these long-term experiments, what is done to correct for drift? And then a couple questions about the drift stage. So can you sort of...
Yeah, okay, so the standard microscope that we use with systems is the Nikon TIE, which we provide that with their perfect focus system. And the use of the perfect focus system is integrated into the software so that the user can just basically specify they want to use perfect focus, and whether they're doing a time lapse of a single focal plane or Z-series across time, or even Z-series at different stage locations and time-lapsing that, the perfect focus system will correct for drift over time.
Okay. So here's a question on the Atlas software, and printing the grid. And so the examples that you showed showed an image already, and so the question is, do you first have to take an entire image in order to be able to prune? And so I guess it's asking.
No, you don't. You don't have to. No, you don't. You don't actually have to acquire a complete preview image. That particular slide was made for a training presentation here by one of our software engineers. Most people, when they set up the an Atlas acquisition, actually navigate around on the screen, navigate around as I showed on the navigation slide, and pick 2 or 3 different locations that they know from their understanding of their sample are going to basically kind of define the a rough boundary of the sample that they want to acquire, and then they click the Create Grid button, and the grid then creates a 2-dimensional grid around that encompasses that area that they, the area defined by the locations they've stored, and includes any the overlap that they've designated between the between the tiles. And then if they wanted to be able to use the pruning function, it probably is going to be most useful if they go ahead then and do a quick preview, which would just acquire quickly without doing any high-resolution stitching the area that they had defined, then they go through and prune the blank areas. And a lot of people are doing 3-dimensional montages, so if they run if they run a quick preview, it'll allow them to prune the area that they don't want to acquire, and knowing that they're often acquiring 20 or more planes at each tile location, it's a significant savings of time to go ahead and do the pruning.
Right. Okay. So I guess the configuration here. Do you have the ability to do photo activation experiments?
Yeah, we do. We didn't talk about it today because we're going to do another webinar on that in a couple of months. There's a secondary scanner that can be integrated into the system that has 2 galvanometers, and then is served by either light from a visible launch or from a single visible laser, or we can also interface fast pulsed IR, i.e. multi-photon lasers, to the scanner for the purpose of doing photo activation. That's fully integrated in with the system in terms of this software modules that control it in terms of synchronizing with time-lapse acquisitions. The other benefit is, I think I'd mentioned in one of the diagrams, there was a polarizing beam combiner that actually allows us to combine the photo activation illumination and the imaging illumination in such a way that the photo activation can occur simultaneously with imaging, so there's no moving parts to switch between imaging and photo activation. So that's especially beneficial in terms of experiments that people might be doing that are photo bleaching or photo activation where there's rapid kinetics involved.
Okay. So let's do one last question here. It's sort of a compound question, and it has to do with crosstalk minimization. So there's sort of some general questions on how a slit can possibly minimize crosstalk and not hurt resolution, and if this has been demonstrated or if it's just a theoretical.
Well, a slit's not going to actually minimize crosstalk. In fact, a slit, when people operate in slit mode versus pinhole mode, they're actually sacrificing resolution in one axis on the XY dimension, and giving up a little bit in terms of axial resolution in the Z dimension, but they're gaining a lot in terms of speed. As I mentioned before, you can run up to, even with the Evolved Delta, which is a 512 by 512 detector, by using a relatively narrow slit or relatively narrow ROI oriented in its long axis with the readout register of the camera, you get up to 500 frames per second. In terms of crosstalk, the crosstalk improvement is going to be seen when you're in pinhole mode, in which case, because it's pinholes, you are gaining resolution of both the XY dimension as well as the best possible axial resolution, and the improvement in crosstalk in a 1D array that we're using compared to a 2D array is going to be significant, which it's at least half of the crosstalk of a 2D array, and can be less, and depending upon the magnification and numerical aperture being used. That's something that, yes, frankly, that calculation is based strictly on optical theory, but it's sound mathematics behind that. We've actually, frankly, never had an opportunity to do a side-by-side comparison with a 2D array with similar specimens. We're basing those numbers that we're giving you on, in terms of crosstalk, on, like I said, on the theoretical calculations. Certainly in terms of axial resolution and in terms of penetration into tissue, we have somewhat anecdotally, based upon people bringing specimens to look at on the system during a workshop and seeing the depth of imaging that we can achieve with the system, and then telling us, well, I can't go that deep with a spinning disc. But again, it's somewhat anecdotal. It wasn't a side-by-side comparison on the same day with the same specimen. But we are hearing that from people, that they like the tissue penetration and the sectioning capability of the system compared to other types of systems they've used.
Okay, very good. And with that, we will wrap up this webinar. And so I'd like to thank all of you for joining us today, and like to thank Jeff for the excellent presentation. Please take the survey on your way out, and if you do have any questions, you can still type them in, and we'll get back to you, or you